Good afternoon. I am Decima Charles. Today, the 27th of April 2017, we are going to discuss inorganic cells. Created by the University of Glasgow is this inorganic cell called each cell, which could be the first step towards creating inorganic life. I am going to tell you about how Professor Lee Corrin and his team of scientists in Scotland has an idea of how to make matter come alive. This may seem ambitious, but when you look at yourself, you look at your hands, you realize you're alive. So that's a start. Now, this quest started 4 billion years ago on the planet Earth. There has been 4 billion years of organic biological life. And as they are inorganic chemists, they make the distinction between organic living world and inorganic dead world. So now, what am I going to try to do is to plant some ideas of how we can transform inorganic dead matter into living matter, into organic biology. But now, let's first talk about biology. Within that infrastructure of biology, we need to remember that the driving force of biology is really coming from evolution. And evolution, although it was established over 100 years ago by Charles Darwin and a vast number of other people and evolutionists, it's still a little bit intangible. When I talk about Darwin and evolution, I mean one thing and one thing only: survival of the fittest. Forget about evolution in a kind of metaphysical way. Think about it in terms of offsprings competing and some winning. So bearing all of this in mind, as Professor Lee Corrin said, they wanted to ask themselves a question frustrated by biology. What is the minimal unit of matter that can undergo Darwinian evolution? This might seem as a profound question, and as a chemist, they are not used to profound question every day, Corrin said. Then he realized that biology gave him the answer, and the smallest unit of matter that can evolve independently is in fact a single cell of bacteria. So this raises three really important questions. What is life? Is biology special? Is matter evolvable? If the question has to be answered in reverse, then when discussing the third question, is matter evolvable, knowing how special biology is also allows us to have an idea of what life really is. Here is some inorganic life. It is a dead crystal. When pressure is applied, it comes alive, pollinating, germinating, and growing. This is an inorganic tube, and all the crystals under the microscope were previously dead. Now it looks alive. Of course, they are not really alive. It's a chemistry experiment that creates a crystal garden. Professor Corrin said when he saw this, he became fascinated because it looked like life. You can see the architecture growing, filling the void, and this is dead. So he said he became positive that if somehow we could make things mimic life, we can attempt to go one step further and see if we can actually make life. But there's a problem, because until a decade ago, we were told that life is impossible to create, that we were the most incredible being in the universe, and that we were the only people in the universe. He said as a chemist, he wanted to know if life was that improbable. He said that he thought that the emergence of the first cell was probable as the emergence of the stars. What if we took it one step further, knowing that the physics of fusion is coded in the universe? Maybe the physics of life is also. But the problem with chemists is that they mostly focus on elements. And in biology, carbon takes center stage, and in the universe where carbon exists, and organic biology, then we have all this wonderful diversity of life. In fact, we have such an amazing life form. It can often be manipulated but has to be dealt with carefully in the lab to try to avoid hazards. But what if we could make matter come alive? Could we have a matter hazard? Think about it. It's a serious question. If our pen and other things could replicate, that would be a bit of a problem. So we have to think differently if we are going to make stuff come alive. And we also have to be aware of the issues. But before we can make life, let's think of what life is characterized by. This is a diagram of the pathway in a cell. The cell is a fascinating thing. Synthetic biologists are manipulating it. Chemists try to study the molecules to look at diseases. You have all these pathways going on at the same time. You have regulation, information is transcribed, catalyses are made. Stuff is happening, but what does the cell do? It divides. It competes to survive. And I think that it is where we have to start in terms of thinking of building our ideas in life. But what else is life characterized by? Professor Curran said he likes to think of it as flames in a bottle. What we have here is a description of a single cell replicating, metabolizing, burning through chemistry, and so we have to understand that if we are going to make artificial life or understand the origin of life, we need to power it up somehow. So before we can really start to make life, we have to really think about where it came from. And Darwin wrote a letter to his colleague where he thought that life probably emerged from a little pond somewhere in some part of the world. But the real honest answer is we just don't know. There's a problem with the origin. 4 to 5 billion years ago, there was a vast chemical soup of stuff. And from this stuff we came. Think about the improbable nature of what I am going to tell you. And just remember, we came from stuff from planet Earth. We went through a variety of worlds. We have the DNA world, the RNA world, and the world's last ancestor. Then evolution kicked in. Now, here we are. Now, there is a roadblock and we can't get past it. You can decode the genome, you can look back, you can link us together by a bunch of cardinal DNA, but we can't get further than the last ancestors, the last visible cell sequence. So we don't know how we got here. There are two options: intelligent design, direct and indirect, meaning God, or some alien life force, but talking about that alien stuff just pushes us further and further from it. The other thing we need to think about is the emergence of chemical complexity. So it's like having some form of primordial soup, and this one has a variety of all 20 amino acids. And somehow this amino acid combined and life began. What does this mean? What is life? And what is the stuff of life? In the 1950s, the fantastic Miller Urey did a famous Frankenstein experiment where they did an equivalent in the chemical world. They took the basic ingredients and put them in a single jar and ignited them and put a low of voltage through. And they had a little of what was in the soup. They had a little amino acid, but nothing came out. There were no cells, so the whole area has been stuck for a while. It got reignited in the 1980s when analogical and computer technology was coming up. Professor Curran said in his own laboratory they are trying to create inorganic life by using many different reaction formats. So they are doing reaction not only in one flask but in tens of flasks and connecting them together. So there's a flow of system through the pipes. With this method, they can do it microfluidly, liquid graphically, in a 3D printer, and droplets with colleagues. And the key thing is to have lots of complex chemistry just bubbling away. But that probably will end in failure, so they need to be a lot more focused. He said the answer lies with mice. With mice, he can generate molecules, then he needs the metabolism, he needs energy, he needs information, and most importantly, containers. Because if he has to get evolution, he needs containers to compete. So if you have a container, it's like getting in a car and driving around saying, this is my car, and I am going to drive around and show off my car. So he imagined that it is the same in cellular biology, emergence of life. So these things together can perhaps give us evolution. So they decided the way to test it in the laboratory is to make it minimal. So what they are trying to do is to come up with an inorganic Lego kit of molecules. It is a very simple kit with 3 to 4 building blocks present, and we can aggregate them together and make it literally thousands of thousands, really big nanomolecules, same size of DNA and protein with no carbon inside. Carbon is banned. With this Lego kit, we have the diversity required to complex information storage. Without DNA, we need to make some containers. Professor Corin said in 2011, in his lab, they were able to take these molecules and make cells with them and put inside them chemistry in the cell. Set up molecules in membrane, in real cells, and it set up a molecular Darwinism, a molecular survival of the fittest. Each molecule is competing for stuff. They all are made of stuff, but they want their shape to win. They want their shape to persist, and that is if they can somehow encourage these molecules to talk to each other and to make the right shapes and to compete, they will start to form cells, replicate, and compete. If they can manage to do that, forget the molecule details. Let's zoom out what this could mean. So there's this special theory of evolution that applies only to organic biology, to us. If they can get evolution into the material world, then they can have a general theory of evolution, and that's really worth thinking about. Does evolution control the sophistication of matter in the universe? Is there a driving force that allows matter to compete? So that means we can start to develop different platforms exploring this evolution. Imagine if we were able to create a self-sustaining artificial life. Not only will this tell us about the origin of life, that is possible, that the universe does not need carbon to be alive. It can use Professor Corin said, we can then take one step further and develop new technologies. Because we can use software control for evolution to code in. So imagine we make a little cell, put it out in the environment to be powered up by the sun. All we need to do is evolve it in a box with a light on. We don't need to use design anymore. We find what works. We take our inspiration from biology. Biology don't care about design unless it works. So this will reorganize the way we design things. But not only just that. We will start to think about how we can start to develop a symbiotic relationship with biology. Wouldn't it be great if we could take these artificial biological cells and fuse them with biological ones to correct problems we couldn't really deal with? The real issue with cellular biology is that we are never going to understand everything because it's a multidimensional problem put there by evolution. Evolution cannot be cut apart. You need to somehow find the fitness function. Corin said the profound realization for him is if it works. The concept of the selfish gene gets kicked up a level and we really start talking about the selfish matter and what does that mean in the universe. Where we are at now is the highest form of stuff. For example, we sit on chairs. They are not alive, but we are. made of stuff. We use stuff. We enslave stuff. We using evolution in biology, and inorganic biology is quite appealing and exciting. We are really becoming very close to understanding the key step in making dead stuff come alive. And again, when we are talking about how improbable this is, remember that 5 billion years ago, we were not here. There was no life. So how does that tell us about the origin of life and the meaning of life? Perhaps for him as a chemist, he wants to keep us from the general term and focus on the specific defining life. We really struggle to do this. Professor Corin thinks if we can make inorganic biology and inorganic matter become evolvable, that would be in fact defined life. He believes that matter can evolve, is alive, and that it gives us the idea of evolvable matter.